The amino acid glutamate is the most abundant free amino acid in the brain and functions as an excitatory neurotransmitter. It is estimated to be involved in most excitatory brain synapses, making it the dominant neurotransmitter used for neural circuit communication. Excitatory neurotransmitters depolarize their target cells, most commonly by opening ion channels that allow flow of positive ions into the cell. Like all amino acids, glutamate has a carboxyl group and an amino group attached to its alpha carbon, as well as its unique acidic side chain. It is a non-essential amino acid, meaning that it is produced from within the body. The neurotransmitter is synthesized in the central nervous system from glutamine by the enzyme glutaminase as part of the glutamate-glutamine cycle. This occurs in the presynaptic neuron or in neighboring glial cells. The isolation and protection of the brain is predominantly maintained by the blood-brain barrier, a system of tissue sites made up of brain vascular endothelial cells, choroid plexus endothelial cells, and arachnoid membrane that restricts and regulates the fluidity of substrates between the cardiovascular system and the central nervous system. Glutamate cannot cross the blood-brain barrier, however, facilitative transporters present in the luminal membrane of the blood-brain barrier actively transport glutamate from extracellular fluid into the endothelial cells of the blood-brain barrier. This high-affinity transport system maintains the glutamate concentration at a fairly constant level. Glutamate plays a major role in neuronal excitability, synaptic plasticity, and immunity, as well as being a key component in behavioral mechanisms like learning and memory. Glutamate metabolism involves multiple reactions, some of which are irreversible. Enzymes that are regulated by activators and inhibitors catalyze these reactions. Alterations in glutamate metabolism are associated with Alzheimer's disease and coronary heart disease. More than 20 glutamate receptors have been identified in the mammalian central nervous system. These have been categorized into two main groups: ionotropic, which are voltage sensitive, and metabotropic, which are ligand sensitive. Each receptor has three types and multiple subtypes dependent on factors such as binding specificity and ion permeability. Ionotropic receptors are fast-acting and can produce large changes in current flow even if the voltage difference across the membrane is small. After glutamate binds to an ionotropic receptor, the receptor's channel undertakes a conformational change, resulting in an influx of extracellular sodium and other ions and an efflux of potassium ions, triggering membrane depolarization in the postsynaptic cell, thereby inducing signal transmission. The three types of ionotropic receptors are NMDA, AMPA, and kyanic acid. NMDA receptors are unique in that they allow sodium, potassium, and calcium ions to flow through. However, for the channel to open, glutamate must bind to the receptor, depolarizing the postsynaptic cell and unblock the magnesium gate inside the channel. Metabotropic glutamate receptors are slow-acting and exert their effects indirectly, typically through gene expression and protein synthesis. These receptors often enhance the excitability of glutamate cells, regulate neurotransmission, and contribute to synaptic plasticity. Memory and learning require synaptic plasticity. This refers to the capacity for synaptic connections in the brain to be altered over time and with experience. The most studied and documented forms of synaptic plasticity are long-term potentiation and long-term depression. LTP refers to the strengthening of synaptic connections through frequent activation. In contrast, LTD is the weakening of synaptic connections. The precise role of glutamate in synaptic plasticity is still unclear. However, it is understood that LTP can occur in a number of ways. The most well-known mechanism is the NMDA receptor-dependent LTP. In this method, the release of glutamate activates the nearby AMPA receptors, but not the NMDA receptors as they are blocked by a magnesium ion. Frequent action potentials cause greater stimulation of AMPA receptors, causing the postsynaptic neuron to depolarize, resulting in the removal of the magnesium blockage, allowing calcium ions to flow through the NMDA receptor. The influx of calcium causes more AMPA receptors to bind to the neuron's membrane, making the postsynaptic neuron more responsive to glutamate, strengthening the synaptic connections. Although acetylcholine is the principal neurotransmitter functioning at the neuromuscular junction of skeletal muscle, Glutamate is involved in modulating cholinergic transmission and plastic changes in vertebrates. The precise localization of glutamate in motor neurons is unknown, and data supporting vesicular glutamate transporters present at neuromuscular junction and cholinergic synapses is contradictory. However, new research suggests that acetylcholine and glutamate are co-released at the neuromuscular junction. This would indicate that glutamate plays an important role as a mediator of synaptic plasticity during muscle recovery after injury or exercise, and may enhance motor function in other ways.